Hi everybody, this was a trailer you might recognize today's lecture about cancer stem cells. Um, yeah, every good movie needs a trailer, of course. This was free of charge. It was a cover of Long Train Running from the Doobie Brothers. Okay, so now come back to science, and I will tell you something about cancer stem cells today, because cancer stem cells have a particular importance for cancer therapy, also using radiation. So I will tell you something about the long-term repopulation potential, what characterizes cancer stem cells, but also healthy stem cells. Then some particular molecular pathways that are specifically activated in cancer stem cells. I tell you something about the relevance of the multilineage differentiation potential of cancer stem cells, which has to do with the plasticity of cells in tumors and also with the potential to metastasize. And also, finally, probably the most important part will be the role of cancer stem cells during the development of therapy resistance in tumors. So first, let's start with normal stem cells. Everybody who is a bachelor in biology or molecular biology or medicine or genetics should be a little bit familiar with the role of stem cells, in particular during the development of multicellular organisms. And all multicellular organisms, of course, they start with a single cell. So in case of mammals and higher organisms, of course, this is a fertilized egg. So in case of all organisms that reproduce sexually, they start with a fertilized egg here. And this fertilized egg, of course, it starts, and after the first few cell divisions, you're talking already about an embryo, maybe consisting of 8 cells. So in the 8-cell status, all the cells are still identical, and a little bit later, this blastocyst develops by orivate differentiation of a single cell layer and forms something like a membrane, and the so-called inner cell mass. And this inner cell mass, I have some problems here with the pointer here. The inner cell mass consists in principle of pluripotent cells. So these pluripotent cells, they can be harvested, can be put in culture, and can be expanded in an unlimited manner, virtually. And when you take these pluripotent pluripotent stem cells, or also called embryonic stem cells, and mix them with a different embryo, and you label the recipient embryo and the donor pluripotent stem cells, embryonic stem cells, differently, then you will later find in the fully developed organism that is born months or weeks later, that these embryonic stem cells, they can contribute to all to all tissues. So this means these embryonic stem cells of the inner cell mass, they are virtually have the potential to differentiate into all 3 lineages of the of the germ layer. So endoderm, ectoderm, and mesoderm. Okay, so these are um, uh, the embryonic stem cells that are relevant for the development of an organism via embryogenesis. You also have stem cells throughout life. So even if the organism is already fully developed, there are still stem cells that are relevant for maintenance of the organism's health. They are important to permanently replace lost cells or cells that are worn out or cells that are damaged. So adult stem cells, they are not pluripotent anymore. It means they cannot differentiate into all 3 germ layers anymore, but they can differentiate only to a limited set of lineages. So for instance, you have in certain areas of the brain, you have neural stem cells. They can still throughout life produce new functional neurons. So in case neurons are lost or damaged, they can be, in certain circumstances, be replaced by cells provided from neuronal stem cells. Probably the most well-known stem cells, adult stem cells, and those that have been studied for quite a long time, are hematopoietic stem cells. They reside in the bone marrow, and these hematopoietic stem cells are of utter importance for the lifelong maintenance of the immune system, and also for the lifelong maintenance and production of erythrocytes, of course. But you also have adult stem cells in organs that were not formerly known to carry stem cells. For instance, in the cardiac muscle, you already apparently have a few stem cells. In skeletal muscle, you have stem cells. They are called satellite cells, for instance. So if you have parts of the skeletal muscle damaged or lost, they can be slowly replaced by cells that are regenerated from these satellite cells, which are adult stem cells. So why we are talking today about, mainly not about the normal cells during embryogenesis or during normal tissue regeneration, but we are talking about cancer stem cells, also called tumor stem cells. Because a tumor that's virtually characterized, malignant tumor, is characterized by an unlimited growth potential, so it can be regenerated even from a few immortal cells. So this means these cancer stem cells, they have virtually, we have to assume an unlimited long-term repopulation potential, or LTRP. So this is also, as most of the famous tumor cell lines that are used in laboratory were generated, so they came from a macroscopic tumor after surgical resection. These tumors were dissociated into single cells, and then single cells were cloned. So there were single cell clones generated, and these single tumor cells that give rise to an entire clone, first in vitro, and were then shown to also produce again the same tumor after injecting them in immune-compromised mice, recipient mice. So these were virtually tumor stem cells, because over many years and decades, these cells that were originally derived from a single human tumor cell still have a complete unlimited potential to grow. So from still in the laboratory, if you have a single of these tumor cells, tumor stem cells from a tumor cell line, you can virtually expand them. You can produce kilograms of new tumor tissue. So that's virtually the criteria when, to talk about stem cells. So stem cells must have this unlimited long-term repopulation potential. When we talk about the difference between normal healthy stem cells in an adult organism and these malignant or cancer stem cells, you have to understand a little bit the kinetics of cell division in these two cell types. So usually in adult stem cells, during life, during the normal replacement of worn-out cells, these stem cells divide in an asymmetric manner. So this is like the stem cells in the bone marrow. Hematopoietic stem cells divide. Yeah, so you have a cell, you have a stem cell here that has virtually long-term or unlimited repopulation potential. It can divide again and again. But during division of these hematopoietic stem cells, normal adult stem cells, two different cells are produced. So one cell, again, is one of the daughter cells is again in adult stem cells, again with an unlimited or long-term repopulation potential, and the other cell is a cell that is either already fully differentiated or is committed to differentiate later after a few cell divisions. And because on each step in this division hierarchy or division pedigree here, you have a split between one daughter cell remaining stem cell, the other daughter cell becoming a differentiated or committed precursor cell. You call this asymmetric cell division. On the other hand, in particular, early in embryogenesis, when the organism is still growing, or later during excessive stress, during wound healing, for instance, the cells under certain stem cells under certain circumstances, they can divide in a symmetric manner. So symmetric stem cell division means one stem cell divides and it produces two new stem cells. Yeah, like here. One stem cell divides and both daughter cells remain fully with stem cell potential. Sometimes later in the division pedigree here, this changes again. It can also become asymmetric again. But globally, during symmetric stem cell division, you see an increase of the number of stem cells or number of cells with long-term repopulation potential. So what you see in tumors, virtually in tumor stem cells, is a mixture of symmetric and asymmetric cell division. Yeah, so you see that when the tumor volume increases, you also, of course, have an increase in the tumor stem cells, and it means the net number or the total number of tumor stem cells must increase. So you must have, at least under certain circumstances, stem cells that divide symmetrically again. So a little bit to the history and who was the first guy who proposed that there exists something like stem cells in tumors. This was actually before the identity of normal stem cells were discovered or were identified. This was in 1875, so there was a physiologist, Julius Cohnheim. 1875 was a big revolution in the ability to microscopically evaluate different tissues, healthy tissues, sick tissues like tumors. Of course, there were a lot of tumors around of which people died. Usually the people had to take material either from animals that were killed for investigation, or from patients that died during from a particular disease. So Julius Cohnheim was very much interested. He studied a lot embryogenesis and the cells during embryogenesis. And of course, what he observed in embryonic tissues was that there are quite a lot of metaphases or mitosis. It's because, of course, during embryonal development in most of the organs, you have rapidly dividing cells. That's why you see a lot of mitosis. In an adult organism, it is extremely, extremely rare that you see a dividing cell in normal tissue. However, when Cohnheim analyzed tumor tissues fixed and put under the microscope, he found a lot of mitosis in these tumor tissues. And by dissimilarity between tumors from an adult organism and tissues from an embryo, he proposed that tumors might be a remnant of embryonal development, so that there's a sort of misregulation or lack of control of termination of the embryonal development that causes later in life a regrowth of tissues that are very similar to embryonal tissues. So this similarity between cells in the tumor and stem cells during embryonal development made Cohnheim believe that between embryonal cells and tumor cells there are some similarities. About a century later, it was possible for the first time to really grow cells from a tumor, different cells, single cells from a tumor, in vitro and let them regrow in a animal again. So these were nude mice. Nude mice are immune. They have an immune defect, so they are unable to reject foreign cells. So that's why they also are unable to reject a human tumor cell. So in the 70s, by this process called xenotransplantation, it was people were able for the first time to show that single cells probably are able to regenerate an entire tumor, a macroscopic tumor that is virtually lethal at the end. Nowadays, you have a lot of different terms that are used if you talk about the stem cells in a tumor. So sometimes they're called cancer stem cells, sometimes they're called tumor stem cells, sometimes they're called cancer stem cell-like cells. Sometimes they're called tumor initiating cells. It all depends actually on the experimental setup or on the observed parameter that you measure. So when you measure the efficiency of regrowth of tumor in a xenotransplanted mice, usually you talk about tumor initiating cells. Yeah? If you talk about the cells that remain after cancer therapy, so which become therapy resistant and give rise to residual tumor growth, you talk about cancer stem cells. They are probably functionally very close links between these different terms, so don't worry too much if you read about cancer stem cells, tumor stem cells, cancer stem cell-like cells, tumor initiating cells, and so on. So, but the first reason why people believe that there are some particular cells in a tumor is the observation of large tumor heterogeneity. So the pathologists or the histopathologists, so which are evaluating the tumor from a patient for the different cell morphology, they know for a long time that most of the tumors, they show large cellular heterogeneity. So there are areas in the tumor where cells seem to undergo more frequent cell division. Other areas of tumor seem to be more differentiated. And also some areas seem to invade into other tissue, into surrounding tissues, and so on. And this tumor heterogeneity within a tumor also was reflected virtually when you took the cells in vitro. So you saw that some cells form colonies, so they're clonogenic. Yeah? So other cells of the tumor just sit there doing nothing or dying out after a while. So there were two possible explanations or hypotheses for this difference within a tumor cellular heterogeneity. One was that the cells just change their biology, their growth potential in a stochastic manner, so that virtually every cell under certain conditions or at certain time points can become clonogenic, so form a new clone and finally a new tumor. And the alternative model, the cancer stem cell model, actually was the idea that you have a large number of cells that are terminally differentiated also in the tumor, which don't contribute to growth anymore, contribute to the phenotype of the tumor, to the disturbance with other surrounding tissues and so on, but actually not to the growth. For the growth responsible are only a very few small subset of cancer stem cells. So how to distinguish between these two possibilities? That was just my method. Probably you have heard about this and used it in the practicum during the course. It's a clonogenic assay or colony forming assay. You have probably done it with some tumor cell lines, which are very easily to handle and to manipulate. But originally, this colony forming assay is done from primary tissues or from primary tumors. So a tumor, when it comes from the surgery, can be disintegrated using particular kinds of mechanical measures or enzymatic digestion, and you get virtually a tube full of disaggregated single cells from a tumor. You can count them, and you can plate them in tissue culture with cell culture medium and with serum and with some growth factors, and observe what happens. And what you usually observe, of course, is that the cells grow as distinct colonies or distinct clones. Yeah, so they don't, if you put, for instance, 10,000 disaggregated tumor cells into a small cell culture flask, you don't see 10,000 colonies that rapidly overgrow everything, but you see only a few colonies. Yeah, but if you wait long enough, these colonies become really larger and larger. Initially, maybe you see them by eye when they are 50 cells large, but you can let them grow and they become 100, contain 100 cells and 1,000 cells, 10,000 cells. You can split them and you will see that these colonies, they have virtually a long-term repopulating potential. Depending on the cell type, maybe 1% of all the cells that you put in have this long-term repopulating potential and form colonies. So if you count the number of colonies here and uh, calculate the percentage from the total inoculated cells, you then get an estimation of the number of cancer stem cells in this tumor. So now the question was um, interesting. When these tumor cells, a single tumor cells, you have to be in mind, a colony, that's what we know for sure, the colony comes from a single cell. Yeah? The experimental setup is done such a way that there are initially only single cells. Yeah? So don't think that the colony, what you see at the colony here, is an aggregation of 100 primary cells from a tumor. No. The colony comes from a single cell. It's a clone that is derived from a single tumor cell. And we believe it's a clone derived from a single cancer stem cell. So what happens when these clones expand, when they grow in size? Yeah, as I said, initially maybe you see them when they are 50 size and diameter and 100, 200, 500,000 cells. Are these all, do they remain all cancer stem cells, or what happens to them? I mean, if from a single cancer stem cell you would grow an entire cancer stem cell colony, you would assume that if you take all these cells and plate them out again, you get a much higher number of colonies then. So if this is done, so you take cells from several of these colonies, plate them out. Unfortunately, we will see that again, there are only maybe similar percentage of colonies growing again. Yeah? So this means that during the growth from a cancer stem cell here in black to a whole clone derived from this cancer stem cell, there's a lot of asymmetric cell division. Yeah? So a cancer stem cell during division usually in most cases does not produce only cancer stem cells during its division. Yeah? A lot of these cells become turned to non-cancer stem cells. Yeah? And that's why you cannot expand so easily the percentage of cells that form colonies. Yeah? So again, if you take 10,000 cells from these primary colonies, put them in cell culture, again, you get maybe again only 100 colonies. So what does it mean? I mean, it's an interesting information if you consider we know today so much about the genetics in cancer, so the mutation, generation of mutation spectra in tumors, how mutations accumulate during progression of a tumor. A lot of mutations, many more than we thought before, sometimes tens or even more of mutations in proto-oncogenes, plus a lot of deletions in various tumor suppressor genes. You could think that, or maybe a cancer stem cell is a cell that just by chance carries a particular set of mutations in its DNA. Yeah. So if this would be the case, of course, because mutations are non-reversible, during clonal expansion, you would assume that all these cells carry the same mutations as the initiating cell, and they would be biologically then the same. Yeah, so they would form a lot of many more colonies than is observed here. But because this is not the case, we have to assume that the property of a cell, of a tumor cell, to be a stem cell, cancer stem cell, is not really determined by particular genetic mutations, but it's determined by epigenetic modifications, and epigenetic modifications are reversible, as you might know. So then, if these alterations that make a cancer cell to a cancer stem cell are epigenetically regulated or determined, we have to assume that the number of cancer stem cells, the percentage of cancer stem cells in tumor is very variable, so it can go up and down. So, and how do we know then how many cells are really important for the tumor to grow? And to answer this question, the most established experiment is a limiting dilution experiment using primary cells from a malignancy and injecting them again in these immune compromised mice like skid mice or nude mice. And by virtually titrating down the number of cells that you inject into the mice and observe how many mice are really, or in how many mice a tumor is grafted, or in this case the leukemia is grafted, you can then mathematically conclude which percentage of um, uh, cells in the tumor are cancer stem cells and are required to form the tumor or regraft the tumor. So this was most frequently done in cells from acute myeloid leukemia from patients. So if you take the cells, acute myeloid leukemia, the malignant cells are in the peripheral blood. Yeah, so you can take the blood is full of malignant uh, uh, leukemic cells, malignant lymphoblasts. They're called. You can count them, and then start with different numbers and inject them into the nude mice and observe how many of the mice, if you have groups say, of 10 mice or 20 mice, and inject a group of 20 mice with the same number of stems, and then you check which percentage of the mice really accept the leukemia and virtually die later from the leukemia, you can get a nice graph. It's shown here. This is, you see it's here in the semi-logarithmic presentation. Yeah, so you have here on the x-axis you have the logarithm of the cell number that you injected, and on the y-axis you have the percentage of mice that develop the leukemia. So you see, if you start here, for instance, with 2 million injected leukemic cells in a mouse, 100% of these mice, if this is a group, so in a group of mice, 100% of these mice got a leukemia and die from it. The same if you go down, the next step, going down to 1 million cells, still all the mice die. Going down one step more, 500,000 cells, still 100% of the mice die. Then if you go further down to 200,000 cells, some mice survive. Yeah. If you go further down, 100,000 cells, 50,000 cells, already more than half of the mice survive. And so survival means they are healthy for the rest of their life. So there is no tumor, there is no late arising leukemia, nothing. Yeah? So from this we conclude that here, for instance, the 2/3 of mice after injection with 50,000 leukemic cells have not received a single leukemia stem cell. Yeah. And if you fit a graph here, it becomes a straight line. Interestingly, this straight line means that the whole mechanism that determines successful or failure of engraftment is determined by the presence or absence of single cells. Yeah. So it's not a curved or a quadratic line here, it's a straight line, and this means presence or absence of single cells. Rare, very rare single cells determines the whole thing. So we have here a point where we have about 67 cells, 67% of all injected mice developed leukemia, and this is the time point by doing some calculations using Poisson statistic. You know that on average in this group you had one tumor cell being injected into the mouse. You had injected altogether 100,000 cells. Yeah, so 1 out of 100,000 cells was most likely a cancer stem cell. So in this case, you know that about 1 out of 100,000 cells, or 1,000th of a percent of all cells, has been a cancer stem cells or leukemia stem cells in this case. Okay, so we finish soon.